Okay, let's look at physiology. So it's very important to think about and self, and it should say non-self, foreign material. So self and non-self. And why would this be the foundation? And your body needs to be on the alert for other organisms because there are always other organisms out there, viruses, bacteria, protozoans. They could try to inhabit your body and feed off of us. So must be vigilant to prevent uh, foreign or foreign. I don't like that word. So non-self organisms from entering. Okay, and that the idea being your body has a recognition of what makes you unique, genetically, making your proteins, special sugars, and whatever is not coming from our body must be an invader, and that's what your body bases itself on as far as what it goes after. So we need to be careful of ethics, especially any kind of human research. And Jenner, who came up with the smallpox vaccine, used children um, to test his vaccination on or his vaccine. He took cowpox and then gave it a little bit of the cowpox virus from pus from cows to kids, and then to see if that would prevent smallpox in people. So you could kind of look at it. Did the child know what was happening? No. So a child cannot decide, to be a part of an experiment that could, kill or severely hurt the child. Can't decide um, to participate. Especially kids back then. I think this was in the 1850s, but I'm gonna check on that, or maybe before. I'm sorry, but definitely kids back then did not have any kind of legal standing like they have now in terms of being able to protect themselves. So um, that's pretty questionable having a child, participate in a experiment like that. And with the parent permission, um, sure, the parent could give permission, but remember, it's again the child that's going to be harmed. So, and even today, that this comes into question when we look at, say, parents have views. This has come up, say, for cancer treatments, usually in religion. So if they're against, their children being treated for cancer and the child gets cancer, they might try to say, oh, well, let's just let nature take its course. We'll put herbs and other sorts of homeopathic remedies and try it. Hope that cancer will get beat back. And oftentimes courts have come in and ordered that the children get standard cancer treatments and therefore, help those children despite what their parents want to do. So an antigen is any non-self. I want to get rid of foreign, but that's what I had there. That can make your body go on the attack. So things like proteins and polypeptides that are found on the surfaces of viruses and bacteria, pollen grains, like people who have allergies, your body's reacting to those perceived issues with pollen getting in your body. Your body is making a big reaction to it. Cancer cells are abnormal. So, and parasites, say like tapeworm, um, your body's going to go after. And The influenza virus has hemagglutinin. And neuraminidase. And we'll see that next slide here. Oh, kind of. Neuraminidase, hemagglutinin. So these are the proteins that these our flu vaccines are trying to be able to recognize and then go after. That's why these you need a flu shot every year, because they mutate and they change, and your body needs to be able to recognize them. So, like with a flu shot, your body gets a small dose of this, gets used to it, has or creates a small immune response, have some memory B cells. If you ever get hit with the real flu, then you're ready to go. You have the memory B cells that can clone themselves and create plasma cells, antibodies, and then go after the flu to kill it. Challenge and response is this idea that we hear a lot of. So your body is challenged and it responds with antibodies and a cell-specific immune response, as well as the various mucus, and you got pH levels on your skin to help prevent the infection in the first place. To make allows the body to make antibodies and respond to these invaders. And you have these macrophages, macrophages that ingest bacteria, and then they take the bacteria, and the antigens that are in the bacteria are kind of flipped and then displayed on the surface of the macrophage or phage. And then helper T cells, they have a receptor that are displayed by the macrophage. You have a lot of helper T cells, only a few can fit the antigen. They bind and then they're activated by that macrophage, and only the helper T cells that have specific binding protein will attach to the antigen. And the helper T cells then go activate the B cells, and they replace a signaling protein. So here we have our pathogen. Nice diagram to show it. Here's our macrophage, and this is our T cell. And then we have signaling proteins activating our B cell. And then we have these right here, antigens. I'm sorry, are antibodies, and I can't go back. So here we have antigen fragments mixing around and then being presented at the T cell receptor on the helper T cell. Okay. So see how plasma cells secrete antibodies, mature B lymphocytes, aka white blood cells, called plasma cells. So you think of B cells and giving rise to plasma cells, and they secrete large amounts of antibodies. Those Y-shaped structures we saw from before. The rough ER makes the antibodies. So these plasma cells right here have a lot of rough ER. You know, those membranes around the. We haven't. Oh yeah, sorry, we did do topic 2 or 1 that talks about the cells. I was thinking I was with the juniors. So you have the rough ER, the membrane with all the ribosomes surrounding it. They're very specialized. So out of all those genes that you have in your DNA, just a few are expressed because they're just doing those few things. So you think about plasma cells being very specialized. Another very specialized cell would be red blood cells. Just a few things, a few genes being expressed. So more on beta cells, they differentiate, develop into plasma cells, which then create antibodies, and they flag cells for destruction. Macrophages right here. So beta cells, plasma cells, boom. All the antibodies, they surround the bacterium. Makes it easier for the macrophages to go chew them up later. These beta cells, these B cells need to create a lot of new copies. The fastest way to do that is through mitosis, of course. They're just making clones, so this is called clonal selection. And this is what happens when you get a flu shot. If you do get unlucky and get exposed, beta cells, these B cells are produced very quickly, and they can slow down, stop the infection, especially if you have a bunch of them. And the antibodies don't live very long, maybe a few weeks or months. This is also when an infant is breastfeeding in the mother's breast milk. There's antibodies that hopefully can prevent infection from various pathogens, at least for a short amount of time. And some of these beta cells or B cells turn into memory cells, which can live long after. And usually they hang out in your lymph nodes, where they can get activated. And if you see it again, you can quickly stop that infection. So here, very nice. We got our pathogen helper T cell. We have the B cell going to the plasma cell. And then that plasma cell making antibodies. Eventually, you're going to have some memory Bs left over. Memory B cells.